Okay, so our next topic with respect to DNA replication has to do with DNA repair. And so we're going to understand what are repairs and the importance between carcinogenic, mutagenic, and actually go through three different types of repair mechanisms that are well characterized. So the first thing to understand is what is carcinogenic and what is a mutant. So a mutant is going to permanently alter the DNA bases. And it's important to understand that most chemicals are not mutagenic. Your DNA is very, very well hidden, it's very well protected, and so it's only a few chemicals that are actually going to be mutagenic. Now, the mutations, of course, can be, for example, an insertion, a deletion, it can be a missense, and missense means that it's either in non-coding areas or it's going to not change the protein. And so your mutations that are going to lead to potential cancer or chemicals that cause this are going to be known as carcinogenics. And 90% of carcinogenics are mutagenic. Okay, but having a mutagen does not mean that you're going to cause a change in the coding regions of your DNA, because many of the mutagens, of course, can be missense mutations. We have a lot of our DNA that is going to not code for proteins, and in that case, they can be, it would not lead to a phenotype, for example. Now, the other type of mutation, of course, will be a substitution mutation. And this is the replacement of a base pair for another base pair. So much of our repair mechanisms are going to be involved with countering these types of mutations in your genome. So overall, we have a general repair of DNA. You have some sort of change. So you have a change in your sequence. And then you're going to have to, your cells are going to have to recognize that change, and then you're going to have to excise the mistake. And most often, this excision is going to be much larger than the actual area that the mistake was in. And so you're going to excise a much larger area. And then you're going to have to repair, and this is going to be via your polymerases, the same ones that replicated your DNA. And then you're going to have to seal, which is done by ligation via ligase. And so in general, all repair mechanisms have this sort of approach to repairing your DNA. Now we have mismatch repair. And so there are many genes involved in mismatch repair. This is not a very simple process. And some of the things that occur during replication are also going to recur during repair. So again, we're going to have to identify the problem site. And then in the case of DNA, you also have to be able to identify the parent or the template strand. So if you have an error, it's very important to fix that error correctly. And if you change the DNA based on the newly synthesized strand, that would be incorrect. So your enzymes have to have a way to recognize the parent strand. Now in E. coli, this is going to be done by your methylation. So in E. coli, during replication, the template or the parent strand is methylated. Now this methylation doesn't last forever, obviously. After replication, it's going to be removed. But in a short period of time after replication, if there is an error, your enzymes can identify the parent strand through these methylated bases. And those methylated bases are shown here on the parent strand. And so it's important because the parent strand remains unaltered and the new strand is changed. So that template strand always stays intact, and that's to, of course, maintain fidelity. And so your new strand is going to match your template or parent strand. Now, some of the things that are going to be similar to our replication is this is going to involve our helicases. So you still have to unwind. And then you're going to have single-stranded binding proteins. And that is going to protect that parent strand as it becomes a single-stranded DNA so that it doesn't have further mutations associated with it. Also going to be associated with DNA polymerase. And for eukaryotes, this is DNA polymerase 3. That's the major replicating polymerase. And then, of course, you're going to have DNA ligase. Now, included with this is a lot of, we'll say, error recognizing proteins, plus some extra enzymes. So there are, for example, and you don't have to remember these, there are proteins called deamethylase, MUT-H, MUT-I, MUT-S proteins. All of them are going to be involved in identifying the actual base or region that needs to be changed. And then those are going to help also to identify the strand that needs to be altered, so conserving the template strand. And so in eukaryotes, this is also very important. In eukaryotes, we don't have, methylation is known not to be the way that our polymerases know which is the parent strand and which is the newly synthesized strand. So it's not really clear how they do that. But one thing is known is that these mismatches are very common in our inherited cancer susceptibility syndromes. And what this is that you're going to pass on to the next generation mismatches within regions that can predispose the next generation to developing cancer at an earlier age. And such mutations that are very well known are the BRCA1, BRCA2 for breast cancer. Retinoblastoma. And there's about 50 known syndromes for humans. So again, these are going to be inherited mismatches, and it's going to predispose the next generation to getting cancer at a much earlier age. Now the next is going to be base excision repair, and this is going to have a damage to your bases, and which leads to a lesion. And so the lesions can be um, inside, or so it flush with your actual DNA. It can cause a distortion of the DNA, so change in the actual um, uh, structure of the DNA. And so, in the case of a lesion that does not change the structure, you have to have specific glycosolases that are going to be able to recognize that lesion and eliminate that lesion. In the case of a distortion that is structural, you just simply have to excise and use an excision endonuclease to cut that, and then you're going to excise it with an excision endonuclease and you form a gap. Now, the difference between the two is, again, the one that looks flush and looks normal, your enzymes have to find that. And so these lesions, again, need to be repaired. So the enzyme class that is going to be able to excise your lesions are our DNA glycosolases. And these are going to be specific for one type of lesion. So they are going to typically recognize the lesions, and a common lesion are going to be deamination of cytosine or adenine. So basically, adenine or cytosine lose an amine group. And then you're going to have your glycosolases remove the base. And so this is going to lead to an A basic site. A site does not have a base. And depending on whether it's cytosine or adenine, it can be a purinic or an A pyrimidinic. Now, after you get excision, you're going to have a gap that's going to form, and this is going to require a DNA polymerase to come in and fill that gap. And then the same story we've been talking about, DNA ligase will seal the gap. So here's an example of a mutation that occurs spontaneously. So this has no outside influence. Your cytosine is going to deaminate to uracil. And so your base excision repair enzyme, your uracil DNA glycosylase, will go through DNA and find these uracils in your DNA. And of course, uracil is not supposed to be there. And so that would damage the fidelity of your, when you have to replicate again. And so it's important for these glycosylases to remove that uracil. And so again, this is a spontaneous process, and it's going to happen greater in single-stranded than double-stranded DNA. So when you have the unwinding of the DNA to expose it, and it can be for either replication that we've just been talking about, or it can be for transcription, making RNA. So whenever you have exposure of those single-stranded DNAs, spontaneously cytosine in your DNA can deaminate to form uracil. And so of course, deamination, it loses that amine group, and it becomes uracil. And so for our DNA glycosylase to fix this, it's basically going to have uracil glycosylase, specific for uracil, is going to find uracil in the DNA. and then it's going to cleave the N-glycosyl bond to generate an A pyrimidinic site. So we get that AP site. So again, here's the AP site. And so there's a base missing. So it's absent of the base. And then at that AP site, you get an AP endonuclease. And that's going to cut the DNA at the AP site. And then you're going to have, in the case of your E. coli, DNA polymerase 1 will fill this in. And then you have DNA ligase that seals. Okay, so you just basically are going to form the new DNA until you get to a nick where you need it sealed, and then your ligase will seal that nick. Now the next one is nucleotide-excision repair. And when you have DNA lesions that are going to change or distort the structure, you're going to require these types of repair mechanisms. And so here again, you have a DNA lesion that distorts the structure. And so this distortion of the structure is going to require the removal of that distortion. Just like all the other repair mechanisms, this is going to be important for the survival of that cell and overall organisms. And in this case, we have our X-in nucleases. And our X-in nucleases are going to hydrolyze the two phosphodiester bonds. On each side of the distortion. So again, it's going to hydrolyze and hydrolyze. In humans, you have the same thing. You have your X-in nuclease, hydrolyze and hydrolyze. And they are unique in their ability to catalyze two specific cleavages. And they do this at the same time, basically, to get rid of that lesion. Now, prokaryotes and eukaryotes do this. The difference is in the amount that's excised. So in eukaryotes, you have a much larger region that's excised, and in prokaryotes, you have a much smaller region that's excised. After you have excision, you then get your filling in. In the case of our prokaryotes, polymerase 1, we'll fill that in. And in the case of eukaryotes, this is going to be epsilon. We'll fill that in. And then you get your nick. And you have a nick over here. And then you have your DNA ligase that will finish the job for both of them. All right, so in summary, all our DNA have repair mechanisms. We're going to have a mismatch repair, base excision repair systems, nucleotide excision repair systems. Our mismatch are going to be, our parent is going to be identified in E. coli with methylated. In eukaryotes, it's not known what is the identifier for our parent strand. Base excision repair is going to lead an apurinic or apyrimidinic site, and that site will have to be replaced with DNA. Nucleotide excision are going to take care of lesions that distort the actual structure. So they're going to remove those bulky lesions, and then they're going to fill them in with polymerase and ligase. The difference between eukaryote and prokaryote is the amount of the DNA that's removed.